Hello and welcome everyone. We just got done in the previous podcast talking about how vesicles formed and how they move to specific targets. In this podcast, we're going to focus on the secretory pathway. We're going to talk about how proteins that enter the ER are going to move in these vesicles that we've already talked about, enter into the Golgi, and then how within the Golgi they will move by vesicles and then be released from the Golgi and then fuse with the plasma membrane to release the cargo outside of the cell. In doing so, I want to spend quite a bit of time actually talking about what happens to that protein in the ER, a little bit of time talking about what happens to the protein in the Golgi, and then talk about two specific pathways of that vesicle going from the Golgi to the plasma membrane. One is constitutive and one is regulated. So that's where we're going to go with the rest of this podcast. So what happens in the ER? One of the things that happens in the ER is that disulfide bonds form. And remember, those form between two cysteine amino acids. This only occurs in the ER. It does not occur in the cytosol or anywhere else in the cell. So this is a very ER-specific reaction. If you see a protein that has a disulfide bond in it, you know it had to pass through the ER. Why are these disulfide bonds important? And why are they put on proteins that go through the ER, the Golgi, and throughout the cell? They add stability to that protein. They make it a little more durable. As they are released from the cell, they may encounter various degradative enzymes or different pHs, and this just makes that protein a little bit more robust, a little bit stronger. And so that's the main purpose, but I also wanted to make a quick note here. I said it, but let's also write it down. This only occurs in the ER. The next thing that happens in the ER to proteins is glycosylation. This is the process of making an ER protein into an ER glycoprotein. It adds a sugar to it. So in the ER, an oligosaccharide is added. So it's not just a simple sugar that's added, it's not just a glucose, but rather it's a string of sugars that are added. And as it's being added, it should be noted that the oligosaccharides added as a chain, meaning this large stretch of sugars are added to the protein at the exact same time. It's not a stepwise. You don't add one sugar, then another sugar, and another sugar. It's the big oligosaccharide is formed, and then it's added to the protein. And this is also only in the ER. You can get simple sugars added to proteins in the cytosol, but to add large oligosaccharides, it only occurs in the ER, and I should also say, and the Golgi, because we'll see in a moment that more sugar gets added to the protein in the Golgi. So why is this important? So let's list some reasons why glycosylation is important. It protects a protein from degradation. It plays a role in holding a protein in the ER until it's correctly folded. They can serve as transport signals. So we talked about the snares, but these oligosaccharides on the parts of the protein can also indicate how it's going to get into the vesicles. If it goes to the plasma membrane, so on the plasma membrane, it helps identify the cell. So that protects it from phagocytosis. It also helps mediate cell-to-cell interactions. Next, let's talk about ER protein destinations. One, they can stay in the ER. Now, if they stay in the ER, in addition to having the ER targeting signal that was needed to initially get into the ER, it also has to have that K-del sequence. So any proteins that are staying in the ER, they have a K-del sequence on them at the C terminus. Now, this is an appropriate time to talk about this. As vesicles move this way with receptors that are originated in the ER, they have to have a way to get back here. And the way they get back here is because those now empty vesicles, because they just released the cargo, have that K-del sequence on them. And that K-del sequence on these vesicles will allow them to fuse back with this membrane. So that's where this very specific sequence comes into play, to bring that ER protein back into the ER once it's left. Now, since we're talking about the secretory pathway, the majority of the proteins in the ER move to the Golgi. A very elaborate system is set in place here to make sure they're correctly folded, because they can only leave the Golgi, they can only leave the ER to the Golgi if they're correctly folded. So what happens in the ER, there are many ER chaperone proteins. So these ER chaperone proteins, they will do a few things. They will help fold protein properly, but they will also bind to misfolded proteins and prevent misfolded proteins from aggregating. At some point, if the proteins never fold properly, the misfolded proteins go to the cytosol for degradation. This is a very specific process. In fact, if just a very minor change occurs, it will not be released for secretion. It will be destroyed. Often, a mutation occurs, and the mutation doesn't really change the function of the protein, but it does change the way it folds just slightly, and the cell doesn't know any better, and so it just degrades that protein. So we often say that this step here is a quality assurance test. It's much more after the quality of the protein as opposed to the efficiency of making proteins or the economics of making a protein. Quality is the number one priority of proteins going through the ER. So we should all say thank you to our ER for their quality control. So now I want to talk a little bit more about those unfolded proteins. And there's this thing that we call the unfolded protein response. This unfolded protein response occurs when there are too many misfolded proteins. Sometimes what happens is too many proteins are being made and the ER can't process them fast enough, and they will begin to aggregate. So too many misfolded proteins, the chaperones can't keep up with them, and they will begin to aggregate. And when this happens, the cell has two responses. Either it will make more ER membranes to help try to accommodate for the excess amount of proteins trying to get processed through the ER, or it may trigger apoptosis, which is programmed cell death. So if it's too much, this is the last resort. All right, the next thing I want to talk about is what happens to the Golgi proteins. When a protein enters the Golgi, what happens? Let me remind you the different parts of the Golgi. The part that's closest to the ER we call cis. The part furthest from the ER we call trans. And these lobes in the middle here we call cisternae. Now, cisternae, you may have one or you may have hundreds of them. It largely depends on what cell you're talking about and how much secretion is occurring. A lot of secretion, you're going to need more Golgi. More processing of the proteins, you'll need more Golgi. So proteins that enter the Golgi, they have one of four destinations. They may return to the ER using that K-del sequence. They may be proteins that stay in the Golgi. You will have proteins then that will go to the lysosome, and that's part of endocytosis. And lastly, the purpose of this podcast is they may go to the plasma membrane for secretion, or to be just part of the plasma membrane, a protein on the plasma membrane. So over here, let's write what happens in Golgi. And the main thing that happens here is that the glycoproteins, those proteins from the ER that contain the oligosaccharides, they are further glycosylated. At each step of the Golgi, they will have additional sugars added to them. In fact, in each of these Golgi stacks, there will be specific enzymes to add different sugar groups to it. So very enzyme-specific parts of the Golgi stacks. Now, the last thing I'm going to talk about in this podcast is how they are released. And your book goes into more detail than I want to go into for the purpose of this class. So proteins are secreted following one of two different pathways. The first is a constitutive pathway. And this is an unregulated movement of some proteins and lipids to the plasma membrane. So some proteins or lipids need to be constitutively or always being processed to the plasma membrane. The second, the regulated pathway, as you might have guessed from the name, is that it's regulated. But more specifically, it's regulated by external signals. So for instance, we could have a receptor here that binds that responds to a specific signal, and then that in return activates this pathway to move this vesicle from the cytosol to the plasma membrane. This is important when we think about how hormones are released, maybe digestive enzymes, or mucus. These and many other proteins that are released, we don't want to be released all the time. You can imagine if you were releasing digestive enzymes all the time, that could be very bad for the lining of your stomach. We only want to release those enzymes when there's a need to digest something. Okay, so that ends this podcast on the secretory pathway. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.